So in this class we shall be talking about mining metabolites from nature, from microbes to plants. I'd like to take you through this talk in four different sections. First, I'd like to give you some examples of mining metabolites from nature. Second, I'd like to now look at what is India's record in mining metabolites. Third, I'd like to ask why should we mine metabolites and how should we mine metabolites. And lastly, I'll give you some take-home messages of how we can mine metabolites, particularly with reference to India. So let me take you through some examples. My first example to now illustrate to you the importance of metabolites in its application would be an example coming from this Japanese lady called Naoka Takahashi. Now, Naoka Takahashi is a Japanese athlete, and she shot into prominence after she won the gold medal for the Olympic marathon in Sydney Olympics that was conducted in 2000. Now, why did she shoot into prominence? Naoka Takahashi was the first non-black woman to win the Olympic gold medal, and when she won this gold medal, people were surprised because until then all the gold medals were won by women who were black or non-white, which were coming from countries like Uganda, Kenya, Jamaica, and so on and so forth. And when for the first time a white woman won the Olympic gold medal, people were astonished. Did she train on anabolic steroids? And so she was checked for anabolic steroids, and they found that she was negative. Curious to find what is the secret behind her winning the Olympic gold medal, an investigator went up to the coach of Naoka and asked the coach what did she train on. Lo and behold, the answer was the athlete Naoka was made to train on the body juices of this insect that you see, that is called Vespa mandarina. Now, this Vespa mandarina, like many wasps that we have in India, has a very fantastic ability to fly non-stop for about 75 kilometers, and it apparently is able to flutter its wings at a stupendous frequency of about 1000 beats per second. All of this it is able to do by a very, very unique mechanism of fat metabolism, the beta oxidation of fat, which was not known earlier. We do not know how the coach of this Japanese athlete came on to know this knowledge in Vespa, but nevertheless, he now trained his athletes, including Naoka, on this wasp body juice. He basically extracted the juice and fed Naoka with the juice. And what you see is that property of breaking fat in Naoka's body, was able to now propel her in the last 5 kilometers or so when you run a marathon. Today, the body juice of this insect is a very famous health drink in Japan, especially if you want to lose extra body fat, because the metabolite in this wasp is able to break down fat faster than we know. Today, if you are a Japanese athlete, you are made to mandatorily take this Vespa drink, and it is said that since 2000, more number of Japanese have won international competition than before 2000, and all of the success owes itself to this modest animal or insect, Vespa mandarina, and the secret it has in trying to break down fat. The second example that I'd like to give you is about the frogs. Now, many of you would have held frogs, and you know they are very slimy creatures, and it is told in the Chinese materia medica, it's been known for the last 3000 years or so, that if you have a bruise on your hand or feet or wherever, the simple thing to do would be to take the nearest frog and sort of soap the bruise on your arm with this frog, and then lo and behold, in 2 days' time, your bruise is completely cured. Now, why is it? The secretions on from the frog's skin apparently has a rich source or cocktail of what is called as peptidal antibiotics. Today, people try to extract the peptidal antibiotic, a type of antibiotic called the bradykinin-related protein, by giving electrical shock to the skin or to the topical surface of a frog, stimulate the secretion and extract the protein out of this frog secretion. There's a whole story about how this was discovered, And apparently Russians knew this much before probably the Chinese, and to now store and not allow the milk to get spoiled, they would drop a frog in the milk can and the milk would not sort of curdle up, and that was the rich traditional knowledge had by the Russians. There's a company in California which is now closed. It is called Magnanin Technologies. Magnanin in Hebrew means shield. So apparently the frog's secretion offers itself a shield from any topical infection, and you know that frogs live in swampy and wet areas where you have a lot of microflora. So obviously the secretions are now trying to provide a shield for the frog against the infection that could otherwise the frog could land up with. Another story related to the frog is coming from leeches. Now you see this picture on your far left. Now this picture is about the feet which is completely infested with leeches, and that happens to be my feet when I was doing some field work in some part of India. And long after the leeches have bitten you and have left you, you will find that your wound has not healed, and this is because of a small chemical that the leeches now put into your skin where the site of injury has been made by the leech, and this is called hirudin, which is a very powerful anticoagulant, and that way the leeches enable blood to flow without any resistance and gulp it up, and once they are full they sort of fall off. Now this technology or this knowledge had been known in many earlier medicinal literature, and even today if you go to certain clinics in London and certain other parts of the world, if you had an abscess in your arm or hand or whichever part of your body, you are offered choice of two treatments. One is to have a surgical invasion to clean up the abscess. The second is now to have a sterile leech put on the abscess. The leech goes on, feeds on that area. As I told, it sort of puts in an anticoagulant, and all the abscess, the pus and other stuff flows out, and your sort of wound is very clean. And of course, once the leech has done the job, the leech is incinerated so that there's complete sterile environment maintained in the surgery. A fourth example I draw from plants, and there can't be a better example of metabolite from plants than quinine, which is told to have saved more number of lives in the world compared to the number of lives that have been lost in all the wars since modern human civilization took its root. Quinine is a powerful anti-malarial compound, and it is told that it has been responsible also for the great British colonization of the equatorial rainforests of Africa to the marshes of Indo-Malaya region. The British soldiers were very susceptible to malaria. Sorry, and the only way they could invade these exotic alien places was to now take a portion of this quinine, and since quinine was very bitter, they would mix it with gin, and this became to be known as a very famous drink called gin tonic, which every British soldier was compelled to take before he set foot into this alien land. And that is how British were able to conquer vast stretches of malarial mosquito infested lands in equatorial rainforest of Africa and India and Indo-Malayan region. My last example to relate the importance of metabolite comes from none other than Waxman, Selman Waxman, who is regarded also after Alexander Fleming as a father of antibiotic discovery. Now he discovered streptomycin from Streptomyces and actinomycetes in 1945, and this heralded the total, I would say, revolution in terms of the discovery of antibiotics. Now this came from his culture of these actinomycetes in petri plates. Any lab in the country can do that. He screened 10,000s of such cultures gotten from soil and discovered streptomycin and of course a number of other antibiotics, for which he of course won the Nobel Prize for the streptomycin discovery. Now why am I relating all of these examples? These 5 examples that you got are from a whole range of taxa, from insects, from animals, from plants, from microorganisms. Now all of these, from the Vespa manarina to the microbe that you saw, are resulting into some sort of product which is enabling in many ways human welfare in very different manifestations, as you saw. These products are often called, or the process of discovery of these products are often called as bioprospecting. You have heard of the word prospecting. We prospect for gold, we prospect for oil, we prospect for aluminum, we prospect for minerals, and so on and so forth. We prospect for coal. When you prospect biological resources, it is called bioprospecting. And so bioprospecting is a way of searching metabolites that might be of some potential importance to ourselves from the biological resources. It could be fungi, it could be bacteria, it could be insect, it could be animal, it could be plant, and so on and so forth. Now, if I have to put the definition of bioprospecting in perspective, it could be said to refer to the systematic search for new genes, new metabolites, new proteins, new chemical compounds, including new microorganisms, so that these are in some way potentially enabling human welfare. It could be agricultural applications, it could be veterinary applications, it could be medical applications. Now, it is no strange fact that if you ponder for a minute and work yourself from bed, as you rise up from bed and later in the night go to bed, you would have unwittingly used a lot of metabolite. Starting from brushing your teeth, you use metabolites which are gotten from biological resources in your toothpaste. You use some sort of beverage to sort of kick you up in the morning. It could be tea or coffee, but if you now look at the tea and coffee, it contains some metabolite. In this case, it contains a secondary metabolite from a plant. Then after you have a coffee, you have gone on to work or to hear a boring lecture like this, but how do you travel to that lecture? You travel to the lecture often with on a bike or a car. That tire is made up of metabolite, in this case an isoprene metabolite, rubber. Later, after the talk, you have a stiff headache, and what do you do to relieve the headache? You again sort of go for natural resources or metabolites gotten from natural resources like the tiger balm, the zandu balm, and so on and so forth, and relieve your headache after the talk. And finally, you had a long day, so you want to now go back home and take a good night's sleep, but then the mosquitoes are bothering you there. Once again, you sort of take recourse to metabolite from natural resources, in this case a pyrethrin coming from chrysanthemum, to now keep the mosquitoes away and enable you to have a good night's sleep. So, if you now look back, it appears from the dawn of civilization, man, metabolite, and nature have been a tripartite sort of relation, where man has always tried to look for natural resources, whatever they might be, and get the metabolite to render the function. It could be cure for a certain illness, it could be for his clothing, it could be for any variety of requirements, end requirements that he or she might want to put them into. And so this has been our relationship with nature and harvesting or mining metabolites. So what I would like to do against this background in the rest of my time is to dwell on what has been India's role or India's contribution in the discovery of metabolites. Now, you just now saw the array of metabolites that we just now discussed through various examples. Has India made any significant contribution to the discovery of metabolites or to bioprospect these metabolites? My answer to this question has been very frustrating, because the answer is not good to listen to. The answer simply is that it is quite dismal that India's track record of mining metabolites from its biological resources has been not something that we can sort of be proud of. Now, all of you probably will be knowing that India is unique amongst the various countries because it has about 4 and now 5 of the world's 34 biodiversity hotspots. Now, hotspots are regions in the world which are excessively rich in biodiversity. And if you now look at the Indian map, you will find we have the hotspot in the Western Ghats, we have the hotspots in the Northeast India, we have a hotspot in the Andaman Nicobar Island, and we have the hotspot in the Trans-Himalaya. Eastern Ghat is not a hotspot, but though it is equally rich, it's not as rich as the rest of the 4 regions. So we have clearly 4 major hotspots in the country. These hotspots are laden with excessively rich bioresources of all sorts. Plants, of course, lower plants, animals, insects, fungi, bacteria, and so on and so forth. But what has happened is we have, as a country, not gotten to the stage where we have extracted in any valuable or value-laden manner the resources that are hidden in these biological resources. And so, if you look at our biological capital, the potential biological capital of our country is huge, considering these 4 biodiversity hotspots. But if you look at whether we are contributing as a in the context of a global contribution, our biological capital, it appears our share is very less. If you look at the patents that our country is now filing from biological resources, we actually rank very low in the number of patents that we can contribute to the global patent pool. So my question is, against this background, against this backdrop, why should we bioprospect our bioresources? Just because other countries are bioprospecting, other countries are discovering new metabolites from bacteria, from fungi, and from plants and so on, why should we also bioprospect? I have 3 or 4 major reasons to justify why as a country we should bioprospect. One is to sustain our own demands. If you go to any pharmacy today and you buy a formulation, if you buy a medicine from there, please look up the place where it is manufactured or where it is discovered. You will find that it will be unlikely that you will find that that has been made from India. The source is some other country. We may be now doing the manufacturing here, but actual discovery was located in some other part of the world. Now the question is, if we want more of these resources, more of the drug, the only recourse is we need to develop and we need to produce that ourselves. And so to sustain our own demand, even domestic, just domestic, not export demand, we need to now have our own sources of metabolite. Second, I just now mentioned that our share in the global market is very less. If we want to now trade in the metabolites as a commodity of trade, as a commodity of commerce, we need to be globally competitive, and for that we need to know what is there in our biological resources. You might tomorrow have a very, very important compound that is an anti-obesity compound discovered from some plant growing in the northeast of India. Imagine how you can now turn that resource into an encashable commodity if someone were to discover that compound and then market it in a manner that it is marketable globally. You become a billionaire overnight, a multi-billionaire economy just from one drug, and that's the power of bioprospecting. Third, and a very important, and this has been echoed in the last 2 decades, is about circumventing intellectual property rights. All of you know what intellectual property rights are or is. Unless we discover, we cannot stake the IPR on the discovery, because if the discovery were to be made by someone outside of India, he or she will have the right to the IPR, not us, though we are the custodians of the biological resource. And we have seen this happening with basmati, we have seen it happening with turmeric, we have seen it happening with philanthus. Now in all of them we had to contest the international court, saying that we knew about it. Unfortunately, modern science was not applied, and so we were on the verge of losing these international patents. It's another story that we won it back after a protracted battle. But why should we enter into such protracted battle if we can discover and apply modern science? Lastly, for students of science, and especially students who are interested in research, bioprospecting has its own unique flavor in contributing to basic science discoveries. Look at the Vespa story, Vespa mandarina story. Now students of biochemistry can use this insect to study what specific beta oxidation metabolic pathway has this insect got. You can therefore enrich the science. Similarly, one can look at what is the role of quinine in the plants. We can ask questions of basic plant metabolism that quinine is doing in the plant, and so on and so forth. So let me give one story. I'll go back to this one. I mentioned about sustaining our own demands. I have a personal story from our lab to now substantiate why we need to bioprospect and why we should not simply keep quiet, simply because we probably are going to endanger ourselves. Let's see if this example strikes you as something eventful. Now in 2009, we had a pandemic of swine flu, and all of you know what swine flu is. It's caused by the H1N1 virus, and it's quite a deadly sort of disease. And the only cure for swine flu then and even now is this drug called Tamiflu. Tamiflu is a derivative of a primary metabolite from plant, which is called shikimic acid. In a 12-step derivatization process, shikimic acid is converted into Tamiflu, and this Tamiflu was the savior of people affected by swine flu until recently and probably even now. The shikimic acid that is used to make Tamiflu unfortunately is not available in abundance, and even today the global supply, the monopoly of supply of shikimic acid is with China, which gets its shikimic acid from a tree, a gymnosperm called Elysium verum. It's often called a star anise because the fruit is star shaped, and the seeds in the star anise fruits contain 2 to 6% of shikimic acid by dry weight. Now this plant is also cultivated, grows naturally. Now in the height of the pandemic of swine flu in 2009, China did something that could not be thought of. What did it do? It now applied a moratorium on the export of shikimic acid outside of its country. So if you are an Indian and if you have the technology to make Tamiflu but you didn't have shikimic acid, you need to reach up to China to import it. Now after the moratorium, China exporters were told, no, you can't export shikimic acid unless and until you take the government's permission. So in a way, the entire world was held at ransom. If you want shikimic acid from China, you better pay the amount that we ask you to pay, otherwise you simply can't get shikimic acid. During this pandemic period, or at about that time, it was fortuitous that we in our lab at University of Agricultural Sciences, GKVK Bangalore, were working on bird flu, which was a pandemic in some way in 2006, 3 years before the H1N1. This is caused by H5N1. Again, for bird flu, the same drug is used, that is Tamiflu, and even then we were worrying about the supply of shikimic acid. So 3 years before the pandemic of swine flu, our lab had been working to look for alternative sources of shikimic acid. It was again a coincidence that in April 2009, when WHO declared swine flu as a pandemic, at about the same time, we published a paper in a journal here called Current Science, where we showed that India has 5 different plant species, all of them yielding between 2 to 5% of shikimic acid from the leaves, and that can become a very important source of shikimic acid to make our Tamiflu. When this story was published, it was now taken up by media all over the world, and what happened is China slowly took back its moratorium, because now you know the moratorium had no value because someone else had already discovered an alternative sources of shikimic acid. The take-home message from this story is, I would say, a moral. Often we now talk about GDP and so on and so forth. Often we talk about the economy. Often we talk about biological capital, as I just now did. But here, this example tells us, forget cashing the capital. Just to save our own skin under dire circumstances like the pandemic of swine flu, we as a sovereign country need to bioprospect our own bioresources so that we can now stand on our own legs and not depend upon a third country for the supply of something as critical as a life-saving drug. And therefore can be one of the most important reasons why we need to as a country bioprospect our biological resources. Now, having justified that, I'd like to now spend some time on how do we bioprospect. It's easy to say that we should bioprospect. How should we bioprospect? Now, we have been working in our lab in trying to organize the methods in a manner that we can now reach up to a search agenda faster than otherwise. Now, look at this flow diagram. In this flow diagram, you have a chain of events happening, where on the top of the chain you have some natural material, you process the natural material, put it through a high throughput screen, and so on and so forth, and finally, if you are lucky, you come to a novel drug lead which becomes a drug at some time later. Now this entire chain you can now look for and decipher for different drugs that have been discovered in the past. For example, Taxol, a very famous anti-cancer molecule discovered by the National Cancer Institute, took about 30 years from start to the end of the chain. Similarly, camptothecin, a molecule that we are working on, took about 28 years from the start to the finish. So it is nearly one lifetime of work that is required to translate the research from the natural resources to the end product. If you look at this flow, one of the most important rate limiting step that sort of conditions how fast you can reach up to the end point is the first step itself of the choice of the natural material. If you were to start with a lousy natural material for your prospecting or searching, obviously at the end of the day you won't get anything, or you have to now spend substantially large amounts of time looking at it. On the other hand, if you now rationalize your natural material in the beginning, perhaps your search will be not only more certain, but also you can now end up the search in a faster time than otherwise. So we try to sort of look at this in a global scale and try to see if we can now in some way devise methods that might accelerate the search of compounds that we want, accelerate the bioprospecting process itself. So if you look at many or most of the research so far that has happened, it appears that it has happened by random walk. You look at a plant, you say, okay, let me grind the plant and do this, do that and all that. Look up at an insect and do something. This is time consuming, also not certain, it is also costly because effort has to be put. Instead of this random search, can we now look at some non-random search algorithms that now can make my search a little bit more directed or targeted, if you so wish. So in this ensemble, we have identified a number of them. I list here 4 of them. For example, it could be, can I now power my search for new metabolites by looking at whether there is an indigenous traditional knowledge associated with the plant already, so that I don't have to make a selection out of the blue or from an empty slate. So indigenous traditional knowledge powered, can it now power search, or can it power or accelerate the search for new metabolites? Second is, can I use a phylogenetic approach to search for new metabolites? I will not be able to now tell you or elaborate on each one of them, but hopefully let me sort of wrap up by telling a few words about each one of them. In phylogenetic approach, what we do is, if I know a certain plant has got a certain metabolite, the question is, if you have the question, can I look for alternative sources of that metabolite? Or in our lab, what we do is, we try to ask in the world of plants, who is phylogenetically related to this plant, and then go to that plant and search only those plants which are related, knowing that phylogeny now in some way has an inertia for the development of chemicals. So chemicals that are found in one plant are likely to be present in other plants that are related to it than those plants that are not related. So that is a phylogenetic rationale. I will not go and elaborate more on this because of want of time. The third and the fourth depends upon what I call ecological knowledge and evolutionary rationale. I will give an example of how this can power search, and this is something that we are in our lab doing for the last few years. Now let us look at indigenous traditional knowledge of how it can power search. The best example that I can give you is a 2015 Nobel laureate's work, Tu Yu, the Chinese scientist who won the Nobel Prize for the discovery of artemisinin. Now she picked up the lead for artemisinin discovery from a book published in Chinese called the Handbook of Prescription for Emergencies by Ji Hong. And in that book, in a certain chapter, was listed the following sentence, that if you take a handful of a herb called Qing Ho, wring it, and then drink the juice of that, is going to cure a person from malaria. This knowledge was taken up by Yu, and then she found that the plant which corresponded to Qing Ho mentioned in this Chinese book of emergency is none other than Artemisia. And the story, of course, all of us know, the world knows, that Artemisia annua, a species of Artemisia, contains this compound called artemisinin, which today is the most important drug against malaria since quinine became to be disused because of the malarial parasite getting resistance to quinine. And this was the discovery completely owing to the indigenous traditional knowledge held by the Chinese Materia Medica. Now, let me now give you quickly an example of evolutionary logic. Now, in evolutionary logic, it's interesting to ask, why do plants have different metabolites, and then go from there to now prospect and use this as a handle to prospect. Take phytoestrogens. Now, phytoestrogens are estrogens that mimic mammalian sex hormone. They are almost similar to the mammalian sex hormone. They also cause the same manifestation as the mammalian sex hormone. Take the example of acetylcholine, which is a important neurotransmitter. Now, acetylcholine is there in plants, and there are lots of plants called stinging nettle, devil's nettle, and so on and so forth that contains acetylcholine. You look at aphrodisiac. Lots of plants have got aphrodisiac. I have always been asking this question: why do plants have acetylcholine when they have no neural system like the animal system? Why do they have phytoestrogen when they don't have a sexual system like that of mammals? And of course, why do they have aphrodisiacs? I feel the answer to these questions is very challenging, but also very intriguing, and I shall come to that in the next slide. Why do plants have all these metabolites? There are two major arguments for that. One argument is that these metabolites have no function in plants. They're simply a spin-off of some basic metabolism. And the second argument is that all of these compounds that you see in plants, acetylcholine, aphrodisiac compound, the compound that we talked about, phytoestrogen, and many, many others, have a deep-seated evolutionary purpose to be there in the plant, only that we don't know. I, for one, believe that plants do have all of these compounds not as a spin-off, but because it now in some way improves the Darwinian fitness of the plants. So, this is very difficult to validate. Many times evolutionary evidences are very difficult to come by, but what we felt in a lab was, if one can successfully address one or two of this and show that plants have a purpose, then it opens up a window of opportunity to bioprospect in a more smarter and a directed manner. So let us look at one example that emerges from our lab. We have been looking at this evolution of laxatives in plants. Now, all of you know what are laxatives. There are a number of compounds. We will not go into the details of this compound. A whole bunch of compounds are known for the laxative function. Laxatives are one that help us move our bowels, and of course it relieves constipation. And the question is, why do plants have laxatives when they don't have a bowel to move? It's very interesting, very intriguing. And so we set up a small hypothesis. The hypothesis simply is that, of course, plants don't need laxatives, but perhaps plants have laxatives in the seeds and fruit to enable the animal dispersal agent of those species concerned to move the seeds or fruits that are taken by them in the gut faster than otherwise. Imagine in the graph that I have drawn, my x-axis says gut passage time, and on the y-axis is fitness. If the seeds are ejected out of the gut of the bird very fast, the seeds are not scarified, and so they don't digest, they are not able to germinate. On the other hand, if the seeds are kept in the gut for too long a time, let's say the bird is suffering from constipation, the seed is kept in the gut for so long a time that when eventually it comes out of the gut, the seed is more scarified than is necessary. The seed does not give a seedling, and so the fitness is zero. So plants would be selected to pack a little bit of laxative compound in each of the seed or fruit, and when the bird takes it, unwittingly the bird takes the pill also, and what does the pill do? The pill now moves the seeds faster in the gut, and once it is defecated out of the bird, the seed is in a proper condition to germinate. And so with this hypothesis, what we now do, we can now make a very neat hypothesis that plants would be selected to pack laxatives in their seeds and fruits to enable optimization of the gut passage time of the seeds in the gut of the birds. Prediction that we can bring out of this hypothesis is, if whatever I have told so far is true, then you have plants of different types, plants which are animal dispersed, plants which are wind dispersed, plants which are water dispersed, and so on and so forth. I would expect that more than by chance, you should expect animal dispersed species of plants to have laxatives in them compared to plants that are not animal dispersed. So from a hypothesis, we have got a prediction. Now we have to give a test of the prediction. So what is the test of the prediction? You can go about doing this, but I won't go into elaborating on how we did. I'll just show you a slide to show you the results. We looked at 114 species of plants that are reported for the laxative activity. 60% of those plant species turned out to be animal dispersed, in a way proving or in a way validating our hypothesis that, look, if you are looking for laxative yielding plant, it is better to go for animal dispersed species of plant than species that are dispersed by all and sundry mechanism. The take home from this is that it's very, very important in the context of accelerating your search for laxatives. The Western Ghats of India has about 6,000 species of plants. Our database shows about 40% of them are animal dispersed. If 40% of them are animal dispersed, about 2,400 species of plants out of 6,000 species of plants are animal dispersed. If a person comes to me, if an industry comes to me and asks me to bioprospect for laxatives, what I will do, I will not prospect for all the 6,000 species. It's waste of time. I will go only to the 2,400 species, and lo and behold, what I would do at the end of the day, after doing that, I am sure to get a positive hit for the laxative yielding plant out of this 2,400 species. So I have actually reduced my search effort by 60%. I don't have to search for 60% of the plants. I only look for 40% of the plant, and with that I am home with success. So what are the sort of take homes? Against this backdrop, what I have just now mentioned, that it's very important to bioprospect. We have shown the justification of bioprospecting for sustaining our own demands. Forget making money. Remember the shikimic acid example and swine flu example. We have also shown that we as a country are very rich in our biodiversity, and we need to also now see how we can use this sort of capital that is vested in us, potential capital that is vested in us. How can we sort of unfurl it? How can we unleash that power that is hidden? The Department of Biotechnology of our country, Government of India, has taken several initiatives in the recent past. One of the initiatives it has taken is to now go on in a war footing to launch programs that involve bioprospecting of all taxa. As you see in this picture, every living organism is potentially useful to bioprospect. It has now explored areas of chemical ecology in partnership with the Northeast region, and it has now looked at a whole bunch of taxa. For example, here you have insect resources which are sort of looked at for their metabolites. There are a whole bunch of stories. In fact, 4 or 5 labs are extensively working on insects as biological resources for bioprospecting. We have already some leads in terms of development of antimicrobial peptides or discovery of antimicrobial peptides from ants. Several other drugs from bugs, as they say. There are people who are looking at amphibians for antibiotics. There are groups in Trivandrum, there is a group in Delhi, there is a group in Bangalore that is looking at amphibians as resources for peptidal antibiotics. There are people who are looking at genes, mining genes from extreme habitat that can now have some important end use, like relieving oxidative stresses in plants. And there are a whole bunch of sort of taxa. In fact, the world is open for discovery in terms of the basic processes and its application in all of this taxa. But my final take home to students is a story that I would like to recall from C.V. Raman, who happens to be, of course, one of India's Nobel laureates in physics. The story goes something like this. He goes one day to Madras, then Madras, now Chennai, and visits his nephew's house, who is also reader of physics in then Presidency College, Madras. And so when he goes to the house, by the way, this nephew of his was just then recently married, and like many Indian bridegrooms, he was sporting a diamond ring on his finger. And Raman, when he saw the diamond ring on his finger, rebuked, and he said, "I say, why are you wasting that diamond ring on your hand?" Now, after that, he had his coffee and Raman left the house. Long after Raman had left the house, this nephew of Raman was wondering, why was my uncle so rude? And then he realized, at that time when this happened, Raman was in the thick of his discovery of light scattering, property of all and sundry objects. He would put anything and everything under the light to see what is the spectral image that he would get from putting or shining light on these surfaces. He did for diamond, he did for rose petal, he did for butterfly wings, and so on and so forth. So what this nephew of Raman did was, he decided, let me now probably do the light scattering of the diamond ring. So he took off the diamond ring apparently from his finger and then used a light scattering device to now study the light scattering spectra of diamond, and for the first time in the world published this work, and that work was the light scattering signature of diamond. He published this work and then sent this paper to Raman in Bangalore, and Raman, when he got this paper, he wrote to his nephew, "Now you have put to good use your father-in-law's gift." Why should I say this? For me, as a biologist, I always look at this anecdote and look at the green forests that dot our landscape, our country, from Trans-Himalaya, that is Western Himalaya, to Northeast Himalaya, Eastern Himalaya, to Andaman Nicobar Island, and to our Western Ghat, the 4 major biodiversity hotspots of the country. For me, this green wealth is our green diamond. And just like what Raman mentioned, what I would like to mention here is, we have prided ourselves to no end about this green wealth resources in our country. We have prided ourselves in the rich ITKs, that is indigenous traditional knowledge, that every village in our country has. But what we have not done, just like Raman's nephew, is we have put precious little resource and effort in discovering the hidden knowledge, hidden secrets, hidden value of these resources. I would sort of conclude by saying, it is time that we stop gloating of our rich bioresources and biodiversity and put them to good national use, such that we can sustain our own demand, we can stake our own IPRs, we can create our own science, we can be globally competitive. In this context, the Northeast India is relatively unexplored compared to the Western Ghat forest of southern India, and through the Northeast region program that is now being operated out of 4-5 institutions in Northeast India and 3 institutions in Bangalore, it offers I would say a very, very rich and unique opportunity to go into the bioresources of the Northeast India and explore the secrets hidden there, and in the hope of understanding secrets first through basic sciences application, and then looking at whether they can have a spin-off in terms of application to societal welfare. So with these few words, thank you very much.